Good afternoon, my dear students. So today we will be doing data collection, data analysis, and how do we display the analyzed data in your reports will be covered under research methodology ETC 3221 module. So I'm Dr. Sangika Nuru as usual. So we will be learning about data representation in general. So now, in our previous lectures, we have already covered how do we formulate a research topic, how to find the research question, how to develop a hypothesis. Then we learn how to plan your research and how to build up your research proposal. So today, after we build the proposal, what we do is we test the hypothesis and uh, we carry on with our research project. So when you carry on with your research project, it is very important how do you collect data, how do you analyze them, and after analysis, once you get the appropriate conclusions or draw conclusions based upon your observations, you can start writing your thesis or your research papers. So in that, you need to display or interpret the data. So this module mainly covers that aspect. Okay, let's see what are the intended learning outcomes of this lesson. So upon successful completion of this lesson, all the students shall be able to understand the different research approaches used in research. Secondly, you should all understand the importance of primary and secondary data and why do we use them. You also should be able to use appropriate data collection methods and data analysis methods when developing your research proposal. Finally, you should use different data display or representation in your final year research project thesis. So these are the intended learning outcomes of this lesson. So now we will move into the lecture. Okay, now in your previous lecture we learned about how different variables are used and how do you plan your research. Now before starting different data collection methods, I would briefly go into different research approaches that we use to collect data. Okay, first one is inductive approach. The second one is deductive approach. Now, when you have inductive research approach, this is a little bit of a high level approach where we aims at developing a theory, whereas deductive approach will be used to test an existing theory. So basically in deductive approach, if you don't have a developed theory using an inductive approach, you cannot do it. So basically, deductive approach is the one that you will be using for your undergraduate projects. We are not encouraging you to go for inductive approach because it needs a little bit of high level research expertise as when you are doing a PhD. So when we talk about inductive approach, usually the inductive reasoning moves from observation to a broad generalization so that it will become a theory. But in this case, in inductive theory, this could be validated, but you cannot prove it because you're taking observations, you may observe the patterns in it, then you generalize it. But in the case of deductive approach, you move from broad generalization to a specific observation. So usually you will uh, start from a theory, then you have observations. Once you have observations, you can prove the theory. So this is what we usually say hypothesis testing method. So what you will be learning here or you will be doing in the final year project is mostly deductive approach rather than an inductive approach. However, in larger or bigger research projects, these both inductive approach and deductive approach can be combined and obtain results. So this is not an uncommon thing, uncommon thing to uh, combine these approaches when you do a research. However, usually under low expertise, we usually go for and recommend you to go for deductive approach. Okay, let's see what is inductive approach again. Now, when you have little or no data exist in literature, you will go into inductive approach. That is why we say we develop a theory where you don't have any theory based upon these observations. So in order to have inductive approach, there are 3 stages. First one is you should have an observation. Second one, you should have an observation pattern. So this is the data analysis part, to find a pattern in your observations. Third step is, based upon your pattern, you develop a theory. We will go for a very simple example. You observe, in order to exist, elephants, they need water. They need water to drink, they need water to wash their bodies, they need water to reduce their heat. Likewise, they need water to exist. This is one example. You also observe there are many animals and plants need water to exist. Okay, you now go to a different experimental setup and observe an observational pattern. So, many animals and plants need water to exist. Yes or no? Yes. So, you all have observed this pattern. So, what is the theory that you can develop? All living beings need water to exist. Likewise, this is very simple inductive approach, but when you come to research, you will be seeing data, you will be seeing observations, you will be seeing patterns in your data. When you analyze it, you will get patterns. Then, based upon the results pattern, you do several other validation techniques to see whether the same result pattern or same property pattern will be given by several different experiments. Then you generalize all these observations into a theory. So this is inductive approach. That is why I said it is a little bit of a high level research expertise. So in order to see patterns in science, in technology, in engineering, in medicine, you need to have a higher expertise than undergraduation. So you will be able to have a better theory. So even the theoretical background and knowledge base, it should be better in order to have an inductive approach. What are the limitations of an inductive approach? Now, once a conclusion is drawn on the basis of inductive method, you cannot prove it. You can only validate it, and also it can be invalid also. Okay, invalidate, when the technology improves, there will be new observations coming, so that the theory that you have developed may be invalid after a certain level of time with the expansion of science. The deductive approach. When you conduct deductive research approach, you always start with a theory. So this theory usually coming with inductive research. So that is why I say when you improve technology, you can either prove an inductive research or invalidate it based upon different newly developed techniques, or whether the original researcher has missed something, you can always rectify it. So this deductive research approach will only start when you have a theory. Deducting means deducing something based upon this theory. Okay, so reason deductively always have a testing theory. So when you talk about testing theories, this means you should have a hypothesis and you should have the hypothesis tested. So this is where hypothesis comes out. Okay, in hypothesis, you predict the solution based upon existing theories. So this is where hypothesis comes. If there is no theory yet, you cannot conduct a deductive research because you don't have a base in order to obtain a proper hypothesis. Now, in this case, a deductive research, you have 4 stages rather than in inductive research approach, you have only 3 stages. Okay, what are the extra ones what we have? First one, you start with an existing theory. Based upon the existing theory, in the stage 2, you will be formulating the hypothesis. Then after formulating the hypothesis, you design what are the experiments that you need, what are the relationships, what are the correlations, what are the validation techniques in order to prove your hypothesis. So then you collect data based upon your design. After collection of your data, you analyze the results. Does the data reject or support the hypothesis? If it support the hypothesis, that means your hypothesis is correct, the theory is valid. If the data rejects the existing theory, so that means there must be certain places that the existing theory is not valid, and you have to give the exceptions for that circumstances. This completely not make you invalid the theory, but you can point out this theory may be valid, but under these conditions, this theory will not be working. So likewise, you have 2 different approaches, inductive research approach and deductive research approach. Based upon which approach you are using, you have to design your research and collect data. So the data collection will solely depend upon which research approach you are taking. Now let's again check an example. Now, same example, a biological life depend on water to exist. The same phrase that was used in the inductive, which was developed in the inductive approach previous slide. We are going to start with that theory. What is the formulated hypothesis? Now you are going for one branch. What is a branch? All land mammals depend on water to exist. Why don't we use all biological things? because it takes so much of time, you will not be able to finish it up. So you take only the part that you can do easily under current circumstances, current financial situation, and your stage of knowledge. So you take all land mammals depend on water to exist. Then what do? You study all land mammals, not birds, not reptiles, not arthropods, just mammals. You take the mammal species and see whether they depend on water. You provide with controls and also you do the experiment. Finally, you analyze all the data depending on variables, independent variable, depending variable, and extrinsic variables. After you analyze the correlations, relationships, patterns in data, you check whether your analyzed data support the hypothesis or reject it. So all mammal species depend on water equals support hypothesis. Okay, if one mammal does not need water to exist, then what happened? You have to revise the theory. Most biological life depend on water to exist except this species. So this is how you do the deductive approach. So based upon how do you approach the research, your data collection methods will be different. Okay, now let's move to data collection. Now data collection is a process of collecting information from relevant sources to find answers to the research problem or to test the hypothesis if you are following the deductive approach. Now once you select which type of data is needed for you to obtain or go for a solution for your research problem, and also to prove your hypothesis or test your hypothesis whether it is valid or not, you need data collection. So this basically collection of information. Now in the case of your research problem, most of these ones will be numerical relationships or some type of experimental based data. So based upon different collection methods of information, the data collection methods can be divided into two categories. Those are secondary data collection methods and primary data collection methods. Now why did I put this secondary data methods in before primary ones? Usually the secondary data methods means that are already been published. So you make use of those data available in different other published sources. These are called secondary. So this is already available. So we put it before primary. The primary ones, that is the ones that you actually collect data, real-time data that you collect in the means of different collection methods. We'll see what are the collection methods we can have for a research. So first we will move to secondary data. As I said before, secondary data is a type of data that already been published. So where will they be published? This could be in either books, previous records. Now in the case of medicine, you always have this epidemiology unit and other units. They annually publish records where you can have access to those data. And also in the case of central bank, every year they put a record or a report. So you can make use of these data or the data available in these reports to support your research problem. And also you can use biographies, also in newspapers, publish census or other statistical data, data archives. You also have internet-based articles, research articles by other researchers in different other journals, and also you have secondary data in different databases. Now suppose you want to analyze an XRD pattern. In order to see which type of a crystallographic unit is present, what do? Go to the crystallographic database, find the XRD pattern where the 2 theta angle peaks come up, and match it up with the database. So you will easily find what type of a mineral you have in your hand. So this is what I'm saying, X-ray diffractograms. You have databases, so you can quickly find out, compare it with the secondary data and obtain a result. So you need these data. If it is cheaper, you can easily access, and also it is convenient rather than you're doing preparation of a single crystalline material, record an XRD, then compare it. It is really hard, time consuming, so much efforts are needed. But if you have a purified version that has already been done and stored in a standard reference database, you can use it. So this is where secondary data comes into it. So advantages of secondary data is mainly it saves time, it saves your effort, expenses of your effort, all will be reduced. And also if it is really necessary, you can do it. If it is not really necessary to do it and compare the results is enough, you can go for secondary data. And also there is an disadvantage. That is, when you use secondary data fully into your research, you may not contribute the scientific society with new data, but you may have different analytical techniques in order to prove your hypothesis. Then no problem, you will be providing a new theory into the society. So either way is okay, but secondary data usage, there is a typical disadvantage that you will not provide fresh data. So once you have secondary data to be used in your research, you need to, it is very important, essential to apply appropriate set of criteria to select which secondary data you should be using. So this criteria plays an important role in levels of increasing the validity and the reliability of your hypothesis testing or your research. And also, when you talk about this criteria, this should include limit to date of publication, credentials of the author, whether the author is reliable, or this paper is coming out of a reliable source where it is coming with a good journal impact factor, journal index is good, or the database is good, database is indexed, standard, everything has to be checked. And also you have to check the reliability of the source where you obtain the secondary data, quality of the discussions, depth of analysis, and the extent of contribution to the text to the development of research area. And there are so many other things, so you have to include such criteria when you select secondary data. Okay, so now what are primary data? Again, I will go back to a few slides before. Primary data means the data that you collect first-hand experience. Through first-hand experience that you collect is known as primary data. Now, when you talk about primary data, they have not been published yet until you do so. And these are more reliable, authentic, and objective to your research so that you can easily use them in your analysis. Okay, when you collect data, primary data by yourself, you will know what are the exact points that you want, what are the exact relationships you need to draw out, then you can collect the data. Okay, so the primary data will be reliable. The next point of primary data is they are still new and raw, so it has not been changed or altered by any other person other than collection. So this validity is very much high than the secondary data, where sometimes the other person has been altered this one, we don't know. I hope in the reliable sources this is not so. So always remember primary data are more valid than. Okay, now let's see what are the data collection methods that are available to collect primary data. First one, you can provide forms, questionnaires, you can make use of interviews, laboratory experimental observations, field trials, and also focus group interviews likewise. So these are the ways that you can obtain primary data. So once you plan your research, you have to develop your own questionnaires, interview questionnaires, laboratory experimental plans, what are the controls, what are the relationships you are going to assess, then what are the experimental conditions, how the experiment is done, and what are the data you need to extract from specific experiments that you do. So for field trials also, you have to determine which type of a place you are going to use, which type of a field for the land area or the extent of area that your trial is doing, what is the wind speed, what is the humidity, what is the light, everything based upon what you are studying. So you have to have the controls, and also you have to go by actual field trials, trial experiments. Then in the focus groups also, you need to determine what are the questionnaires, what are the discussions you need, what is the plan. Once you plan, you collect the data by providing different means. So questionnaires, you can have a better sample. Okay, you have a good representation, sample design. Provide the questionnaire to the people in your sample. Interviews also the same. You find the describe your study setting, what is the population, to which type of a people you are going to use, which educational level, whether you are going to have different correlationships. So all these things are changed according to your research plan. You will be distributing all these things and collect data. Once that is how you do it, primary data. In the secondary data, you can collect through oral histories, combination of research, online tracking, online marketing analytics, and social media monitoring. So these are certain type of secondary data collection methods. Here you don't have the control over the variables. You can get the data extracted from different sources and check whether there is a pattern with what you are expecting, what you are checking, and there is a correlation. So based upon the way you are doing, these are the data collection methods available for different types of research plans you are doing. Now, in the case of questionnaires, interviews, focus groups, these also can be qualitative as well as quantitative research. Now, in the case of qualitative, you can go for human behavior questionnaires and also focus group interviews, even laboratory experiments that I have said here, that could be case studies and also different types of patiental observations. Likewise, depending on what are you studying, you can go for different primary data collection methods as well as secondary data collection methods. Now, this secondary data collection methods through social media monitoring is rapidly growing because for most of the time when you come for IT based research, you will be using secondary social media monitoring data as an analytical tool in order to find whether there are correlationships existing. So this is how you collect data. Okay, now once you collect data based upon different methods, you have different sets of data based upon what type of a research you are doing. First of one is observational research. Okay. In observational research, you observe data through observations or behaviors or activities. Observations based upon behavior or activities. So usually the methods involved are human observations, open-eyed ended surveys, and also you can make use of instruments or sensors to monitor and record the information. Now you can see all these data type of research is based upon observations. You have a photograph, you have a micrograph, you have the SCM images, you can have a microscopic images. Now you can see the COVID microscopic image in the left bottom corner. So this are observational data, and also this scientist is observing what are the differences that she found based upon their treatments, whether the stem is coming in different colors or whatever the things that she needed. Second type of data are experimental data. Now this experimental data are collected through active intervention by researcher to produce and measure changes to create differences when a variable is altered. So usually what it means is you change a variable keeping all the other extrinsic variables constant and check what happened to the y value or depending that variable. So as you can see, the temperature is varied in time between 0 to 120, and the conductivity was measured, and concentration of something, some ion generation is also measured, and also some other quantities are measured. So these are, once you change one parameter, how do the other parameters change? So this may be a reaction mechanism or what, something based research, but I'm not sure, but this is experimental type of data. In the second or the below diagram, it could say 1 over C, that means the concentration, inverse of concentration is used as the independent variable, and rho that is conductivity as the dependent variable, and plot conductivity based upon 1 over C. And 1 over C is mathematical expression. So this is some raw data converted to a theory. To prove a theory, this has been done. So this 1 over C is linearly related to conductivity, has been measured. So likewise, you alter what are the data, the variable is altered, and measure the changes in y quantities or variable dependent variables while keeping all the other extrinsic variables constant. So this experimental data usually allows the researcher to determine a causal relationship, and it's typically projectable to larger population by altering the variables. And usually when you talk about the experimental data, they are often reproducible and also expensive. Okay, reproducible means if you give the exact same conditions, the experimental data should give the same pattern with the same final results. So this is called reproducible. This is one way of validating your experimental data. If your data could be reproduced, your relationship could be reproduced, then your data is valid. So you have to remember this. Reproducibility of data is one of the ways that you can prove whether the data is valid. The third type of data that is collected based upon the type of research is simulations. Now when you do a simulation, you generate the data by the operation of real world process or a system over time using a computer program. So you call it computer test models. So once you give the test model, you will then generate data. So the test model plays the key role in this one. So simulations will determine what could happen under this certain condition. So I have given you two different examples. First one is a SolidWorks based experiment for stress generation in a tire when it reaches the floor or when it touches the road, the stress generation that is simulated and obtain the results. The second one is weather parameters are simulated and observe the patterns based upon the data that collected from the satellites. So these two are examples for simulation data. Other than that, you have already done 3D SolidWorks and all, so you know how to do simulation data. You might be knowing what are the parameters that you can measure using simulations. Okay, the fourth type of data that could be collected based upon the different types of research methods you can use are derived or compiled data. Now, in the case of 1 over C that I have found is also a derived data. Once you run a set of one experiment with different conditions of different x variable changes, you can derive another set of data, like if the linear relationship was observed between x and y, and the slope will give rise to another quantity, and that quantity is again you're going to plot with temperature or something. These are called derived ones. So usually in research you may use such type of data. Okay, you are not going to use the raw data as it is, but you will be derived based upon different transformation, whether it's going to be arithmetic formula or a aggregation. So you can use different techniques. As I said, you can use interpolation, extrapolation of XY based graphs, and also you can make use of the slopes at different points. Likewise, you can have different derivation types of data to be used for the second study. So you can call them as derived or compiled data. So these are the 4 types of data that you can collect based upon the type of your research that you do. What is the data type? Observational type of data, experimental data, simulation data, and 4th one is derived or compiled data. Okay, so once you collect data, you have to analyze those data in order to go for a conclusion, valid conclusion. So in order to go for a valid conclusion, you need to analyze all the data to find different patterns, whether you are going to derive different data and again check the patterns. This analysis is the most critical part of your research. And the analysis of data will summarize all the patterns of your collected data, and also analysis will enable you to go for different relationships and patterns. So this data analysis involves interpreting data gathered through the use of primary data collection methods as well as secondary data methods, using analytical methods, using logical methods, and also you can use patterns and relationships and trends would be obtained or extracted based upon your analysis. So this analysis could be descriptive. That means the analysis could find what had happened. Exploratory. In exploratory, it says how to explore data relationships. What happens? What are the relationships that you can draw out? Then diagnostic analysis. Why it had happened? Then predictive analysis. What will happen? That is what we showed in the simulation in the second example. The weather patterns. What will it happen based upon the results that you found? Then prescriptive analysis. That will tell you how will it happen. So these are the basic types of data analysis and as I said before, depending on the approach of your research, whether it is inductive or deductive, and based upon your type of the research, the analysis techniques will be different. They will include different theories. Based upon the theories that you do, you can derive relationships from X and Y. And these relationships should be elaborately written in your thesis. So this analysis could involve image processing, statistical methods, and simulation methods in order to go for conclusions. So these methods will basically depend upon what you are doing and how you are doing. So I will not go in detail because we are having diverse focus areas. In diverse focus areas, you will have different analysis techniques in different areas. For certain ones, you can go for X and Y relationships, and also you can make use of mathematical or scientific equations in order to get the relationships to be proven. Or in some areas, you can have statistical methods like regressions, correlations, t-test, uh, and all types of standard deviation, means, and all could be used in data analysis. So I will not go in the depth for you, because this analysis will solely depend upon your research project and your title. So mainly the analysis part you will be taught according to your study in your final year during your research project. So if at that time you have a problem, you might come and ask me, what can I do? How should I help you, or how you need my help? So at that time you can ask. So basically, this is most critical part, and you have to find what happened, how to explore the data relationships, why it had happened, what it happened, and how will it happen. So it all depends upon what you're doing. Okay, now in order to have this analysis, we'll see what are the advantages of primary data and disadvantages. Now, in the case of primary data, usually the researcher or the investigator collect specific data based upon your problem under the study. Okay, what are the experimental conditions you should vary, what are the circuit components you are using, what are the temperatures, what is the humidity, if it is, whether it should be a void water, whether it has to be operated inside water. It all depend on the data that is related to your problem. So investigator could always change the variables, extrinsic variables, depending on what you want to prove. So there is no doubt about the quality of the data that you collect for yourself. So the data that you collect will always be the best quality for you. And also, if required, it may be possible to obtain additional data during your study period in order to prove your hypothesis. So this is the basic advantages of primary data usage. So what are the disadvantages? Now, in the case of primary data, the investigator has to conduct with all hassles. He wants to do all changes and all experimental planning, cleaning, then processing everything. Decide why you are doing, what you are doing, how do you collect it, when should to collect. Everything has to be considered and planned by the investigator. And also, how do this data is collected, whether you go personally and collect, or you put certain people to collect, or you are going to the phone and do the phone interviews, or you use internet interviews, whatever the mode you are using. And also what type of a funding, whether you have enough money, and whether the funding agencies are providing with enough money, and what all the funding agency requirements has to be managed by the investigator. And also, when you collect data, you have to get the ethical considerations like consents, permissions. If you are using people, you always need ethical clearance. If you use animals in testing, you need ethical clearance. Likewise, you need ethical clearance. And all these hassles has to be contended with the investigator's work. The second disadvantage is whether your data collected are of high standard. You have to ensure the standard of your data, whether all the desired data is obtained accurately, and the data is obtained in the format that is required. Now, this is very crucial when you're designing a questionnaire. If you don't design a questionnaire very carefully, the format of your data might go here and there, and you will not be able to go for the solid conclusion. And also, especially when it is questionnaire type or people is involved, whether there is fake or cooked data is there. Okay, somebody has altered the data. No, you cannot do it. So you have to ensure that your data is quality. And also you should remove unnecessary useless data which has been included by mistake or something. So all these hassles should be conducted by the primary data. And always collection of primary data involves high cost. So this cost obtaining data is often become a major problem in many studies, specifically for interviews, focus groups, questionnaires and forms, and also in experimental data generation, you need to pay different agencies for different testings to be done where you don't have it in your institution. So always cost has to be managed by the investigator. So this is the going to be really nuisance to certain extent. So these are the disadvantages of using primary data. Now, why secondary data is important? Now, when an investigator has a difficulty in obtaining primary data under expected conditions, you can go for secondary data. For example, now in this year you have a longer drought season, but what you want is actually heavy rainy season in order to get the rain data. But this year you have very low rainfall. Then what do? You go for a secondary data that is already been published elsewhere with your relevant expected conditions. If it is very much difficult, you can go. Okay, you want to take the gravity values in moon at different times. What should you do? Can you obtain it here? No. You go to the specific library, specific database, get the answers there if it is available. And also, when the primary data does not exist, you can go for secondary data and then you can derive data. You always could derive data. And when primary data is present, but the respondents are not willing to reveal, then again you have to go for secondary data that has already been published or that had already been done in another setting. You can make use of such data. Okay, now let's see what are the advantages of using secondary data. Now, in the case of primary data collection, I said the investigator has to have a very lot of work there. So, like monitoring the funding, where to obtain the data, how do you do the collection, whether you are personally doing it or whether you're going to hire people, how do you manage salaries, how do you print them, how do you do all distributions, transportation, all hassles has to be maintained by the investigator. But in the case of secondary data, somebody else has been done all these things, so you can just collect the data from the published sources. So there will be no hassles in collecting secondary data. So there will be no hassles. It always have a less cost than primary data. So we can say the secondary data is less expensive. Now, in the case of primary data, the investigator has personally ensured that the data is quality, whether they are going to be in order with your research or the study. But in the case of secondary data, the investigator is not the person who is collecting data, so the investigator is not personally responsible for the quality of the data once they are when they are published. So these are the advantages of using secondary data. In the case of disadvantages, the data is always collected by a third party, not by ourselves, right? So when you collect the data for yourself, or your data will be reliable, you can make sure, assure that the variables are in controlled manner. You know all the conditions that you have taken. So the reliability of the secondary data will always have lower value or lower reliability than the primary ones. So when you make use of secondary data, the accuracy of your results will go down. Second disadvantage is the data collected in that use as secondary data is from another location that another location to that of your interest. So this data that is collected in another location may not be suitable for the interested area due to variability of the environmental factors. For example, if you want to find the internet addiction in Sri Lankan teenagers, and the teenagers did not provide you with answers, so you go and take the internet addiction results of a European country of same income and all, but the data will be having specifically a lot of influence of culture, and also the seasons effect will be there. So the data that has been collected and what you're going to make use for your system, for a system that you are developing, may not be same as the ones that you are interested. So the variables in the environment may have not suitability of secondary data. So you may not be able to use it, or you may need to alter it, or you have to put a assumption stating that this and this is equal. Then the third disadvantage is when the secondary data they will be obsolete and very old with the passage of time. That could be due to technology improvements or some other means. These data might not be valid for the setting that you are doing your research. So this is the third disadvantage. Fourth disadvantage is the secondary data can distort your results in your research. So you may need to amend or modify them into your current conditions, so that there will be uncertainty inserted by the secondary data. And also secondary data may raise issues of authenticity and copyrights. So you might not be able to use them also. So these are the disadvantages and advantages of using secondary data. Next one. Okay. Now in the case of your field as a technologist, most of the time your experimental Now we'll move to experimental data collection methods. Being a technologist, most of the time we think you all be doing experimental based research. So the data collection via experiments will be of prime importance for you. So prime method of inquiry in science is experiments. There are key features. You have control over variables. You can carefully do the measurements. You can make use of different instruments to improve the precision and accuracy. And also you can measure based upon different conditions that you make. And also you can establish a cause and effect relationship between the variables in a experimental data collection methods. Usually in an experiment, the independent variable is manipulated and dependent variable is measured. Now in the case of any extraneous variables are available, then what you do is you make them control. Now here you have two parts. One is the control part and one is the experimental. Now what you do is, if you want to check the fertilizer, effect of fertilizer, you provide all the other factors like water, sunshine, the size of the pot, soil should be all identical. Only exception is for the control you don't add the fertilizer, and for the experimental ones you have to add the fertilizer. If you are going to vary the fertilizer amount, you can have many experimental setups and you can move with different experiments. Now here you have plant growth is dependent variable. Then you can measure the days taken to flowering, the height of with respect to. Okay, now let's see what are the three different types of experiments that are available under experimental data collection methods. First one is laboratory, or we call it controlled experiments. This is not only for biology but also for our engineering sciences. We use laboratory and controlled experiments. Now this one we have a experimental conducted in well controlled environments. You know what are the variables that you change, what are the variables that has to be kept under control, and what are the things that you need to observe, or what is the dependent variable. So this laboratory or controlled experiments, they are not necessarily be a laboratory, but it is under controlled variables. So therefore, since all the variables are under control, you can obtain accurate measurements as much as possible in this controlled experiments. The second type is field experiments. Even though you are doing engineering work, you need this field experiments. Field experiment is done in a real life environment. So you don't have the control over variables as much as you do it in the laboratory, or what we call as controlled experimental setup. So this experiment still the investigator manipulate the independent variable, but it is usually under real life setting, so cannot control most of the extraneous variables. Third one, you do it under natural experimental conditions. That means under exact condition where your application is running. So the environmental participants will not, you cannot change them, and there will be no control over independent variable as it is in the other two. Okay, now once you collect data, you need to check whether your data or results that you obtain through primary or secondary data are reliable. This is one of the key importance of collection of data. When the data or result is repeatable, we call the result is reliable. Okay, if you do a set of experiments again, the same conditions, you should be able to get the pattern, same pattern, same repeatable data, then you have higher reliability. Usually, this reliability relies on the accuracy and the precision of the measurements and also the instruments that you use. So, always you have to have calibrations running, and also you have to make sure your instruments are well, give accurate readings, and how precise your reading should be in the case of deriving results. So, if you need high precision data, you always need to go for sophisticated instruments. If you need low precision data, you may use less sophisticated instruments. So whatever the instrument that you are using, the reliability of your results will be affected. Okay, the second point is whether your results are valid. Now, validity usually refer how accurately the method you use to measure is intended to measure. Okay, so if your method is valid, most probably your results will be valid as well. So if the research has high validity, that means it should produce results that corresponds to real properties, characteristics, and variables in physical or social world. So in the case of social world or physical world, if you can interpret the results accurately, you can have high validity. High reliability is one indication that measurement is valid. Okay, reliability is only one measure for validity. If a method is not reliable, it probably won't give you valid data. Next thing you need to consider is the credibility of your research. Now this is coming to the research now. Credibility is whether the research is trustworthy. Is your research believable? So the research conclusions will support your research findings. Now here, this is talking about your overall research and overall conclusion. So if you find a relationship between one variable and the two variables, you have to prove the reader that this relationship is true in another two methods, two independent methods, that is called triangulation. Using different data collection methods in order to check the consistency of the finding, you get triangulation. For example, you want to say some filler reinforces the material that you are preparing. How do you say? You do the tensile test. Reinforcing, tensile strength should be increasing. Second one, you find the Young's modulus. Young's modulus should also increase if the strength increases. Another way, then you take tear strength. The material tear will be high. Under high load only, the tearing will occur. So you have three different data collection methods. But if you can make use of a different equipment, for example, dynamic mechanical analyzer, but you can measure modulus, even that one is giving high results, then it is a second independent method. Then you can find the reinforcement using the dispersion of the filler inside the material. Then you use a micrograph or photograph or some dispersion index measurement, then that one will also prove that dispersion is better. So there will be higher bonding. So likewise, if you have different independent methods proving the same results, then you say your research finding is credible. Second way of assessing the credibility is member checking. The data, interpretations, conclusions are shared with the participants and allow the participants to clarify what their intentions were, correct the errors, and provide additional information where necessary. That is what we are doing under publication in peer-reviewed journals. So in peer-reviewed journals, that means once you do the research, you write the research journal article and submit into a peer-reviewed journal, then the editor will read your manuscript. If the manuscript is worthwhile to be published, he is sent to 3 different experts in 3 different areas of the world and ask them to read and provide feedback on your paper. So they will read and check whether your research finding is really credible and sufficient data analysis techniques are given, sufficiently proven the hypothesis. If not, they will ask, this person has forgotten to prove it in another method, experimental method. So please provide this experimental method if you want to publish it. So that means you have actually forgotten it. So in that way, you can get member checking and get the credible research finding. That is why we say always read research articles from peer-reviewed journals, not in the money paid non-peer-reviewed journal articles. So always you have to be credible of what you publish. Okay, so once you interpret your results, make conclusions, when you are writing your thesis, when you are writing your the papers, you need to display your data and results in a way that your reader can understand and catch up all the points very easily. And also you have to provide your results in extensive and comprehensive way so that the reader would be getting all necessary information from the publication. So this makes data display or representation is important. So in order to display your results or data, you can make use of tables, you can make use of graphs. These are the 2 main data display methods that could be used for scientific writing, either tables or graphical format graphs. Other than that, nowadays you can use graphs, micrographs, diffractograms, then chromatograms. Likewise, you have different other data output methods that you can directly use as the data or results display methods. Okay, now when we talk about the table, when you put a table into a publication, that means in the thesis or your paper, there has a table structure. Okay, every table should have a number starting from 1 to the final one in the order of appearance. Okay, in your This is when you insert a table. Remember, the table number comprised of both your chapter number and the sub number. So chapter number, if the table appears in chapter 1 and this is the first table appear in the chapter 1, your table will be table 1.1. If your table is chapter 1, second table, the table number will be table 1.2. First digit will be chapter number. Second digit is the which table is appeared in first table, second table, third table. Likewise, you have a table number. After you putting the table number, you put a dot and you have to insert a table title. It should be the description of the data provided in the table. Now this table I have taken from one of the research articles in Wiley, and you can see the third one is column headings. If you have two properties, you can have column headings and subheadings. And the purple color box, it is giving row headings, or it is also called stub. Okay, that is row headings. This one is data field. Now, if you have a detailed table like that, you have, as I say, here in this table, you have table 1-1, that is the first table under chapter 1 is table number. So first digit 1 is for the chapter, second digit after the dash is the position of your table. This is the first table under chapter 1, that is 1-1. If it is 10th table in chapter 1, 1-10. If this is first table in second chapter, you will be writing table 2-1. Now here the table title, it says characteristics of study participants with and without incident diabetes by sex. So it describes men and women. So in men, this is called spanner heading. You have column subheadings with incident diabetes, without incident diabetes, and p-values. Then you have raw headings like age, biomass index, steatotic blood pressure, diabetic blood pressure, and also marital status, whether it is single, married, or divorced. These are raw subheadings. And you have a data field, and also if you have any important data to be put as footnotes, you can have like values, a mean with the standard deviation. Likewise, you can have a table. This is the detailed table structure. The previous one is the basic table structure. Okay, now second method of representing data is graphs. So graphical representation is more easy to understand. The analyzed data you can easily predict, you can easily see the patterns, and also you can easily abstract what the researcher or investigator or writer is telling. Now in a graph you have a horizontal axis which is called abscissa, and which is commonly known as x-axis. The vertical axis is called ordinate, which is commonly known as y-axis. So usually the x-axis or abscissa represents the independent variable, and y-axis or the vertical axis or the ordinate represents dependent variable. The intersect, this is not intercept, you call intersect of x and y axis, intersect at either 0 point or 13 point. Now this is an example of graph that you can put in the thesis. Now, when you insert graphs, remember, similar to tables, you should always have a title describing its contents. So here, again, you have to include figure 1, or that is again, you have to include figure 1.1 or 6.1, or anyway, along with the chapter, you have to indicate the figure number. And always, the title of the figure should describe its contents. Then, the axis should be drawn in appropriate rational scale. And always remember, you should label your axis properly and clearly along with the units. For example, number sold. Do you have a unit? No. Day? No units. But in the case of second, the bottom graph, you have time. Time, of course, you have a unit which is day. So within brackets, it says time measured in days is given in the bottom right. That means x-axis. Whereas the bottom graph, y-axis is weight loss, which is the percentage. So the percentage is given in brackets. There is another way of providing units, that is, instead of putting brackets, you can put the division sign after weight loss, put the division sign and write the percentage, then it will say that is the unit. So that is one way. Okay, so these are the types of graphs you can use in scientific writing in order to express your results in thesis or a paper. You can use histograms and bar charts. Now, the main difference in histograms and bar charts is, in a histogram you don't have gaps, and in the bottom you have number ranges. In bar charts, you have categories rather than the number ranges in the x-axis, and always you have gaps in between the bars. Then the other graph type you can use is line graph. This could be either relationship plotting, or you can just show the line graphs. Then you have stacked bar charts, pie charts, and also point charts. Now, usually in the case of line graphs and point charts, that is also called dot plot, if you have a relationship, mathematical relationship between x and y, you will draw a line. Other than that, you don't draw a line graph, but you plot the data points as a dot plot or a point chart. If only you are having a relationship between x and y, you can draw the line. Okay, the next important point in graphs is you should include error bars that enhance the visualized variability of the plotted data in a Cartesian graph. So if you have a bar chart, always remember to put this error bar. You have the data point, and the error bar indicates the estimated error of this measurement. If it is a dot plot, you put only in the y direction. If there is an error in both x and y directions, you can put the error bars in both sides, x direction and y directions, in a scatter plot. In line graphs, bar charts, dot plots, all the y is the variable. So you will express the bar chart in only the y directions. Usually, this error bars display either the standard deviation of that measurement, standard error, confidential interval, or the minimum and maximum value ranges in the data set. So these are the ways that you can put error bars in your graphical results representation. So always remember, it is important to put the error bars in your report. Okay. Now let's see what are the other methods of putting results in your thesis. You can put SCM micrographs. If you're doing nano type of research, you can always use scanning electron microscopic images as this one to be included. Then you can put the TEM image. The TEM image is also could be used in your thesis. And also you can support your results with the photographs. Now this photographs will also show you how your results vary, what happened with different treatments. And remember, when you put any figure, any graph into the thesis or a paper, they should have a figure title and the figure number according to the sequence they are appearing. So as I stated in the tables, again the figures should be numbered according to the chapter. Within the chapter, the position of the figure should be the figure number. Then always remember to write the figure title meaningfully so that the reader would observe, grab all the necessary in it. Okay, so let's see the summary. So today we learn about two different approaches of research, that is inductive approach and deductive approach. Based on inductive approach and deductive approach, you have to design your practical set or experiment set. Then you can make use of primary data, which is what you really measure and generate within real-time data that you generate when you do the research. Then you can also use secondary data if you are in a position that there are no primary data available. Then we learned how to analyze it and why it is essential. And as I said, the data analysis will be based upon what type of data, what type of research you are doing. You can make use of statistical analysis, simulations, and all sort of other data processing techniques that is relevant to your topic. After analyzing, your results could be has to be represented or displayed or presented to the reader. So how do we present the data? That is either in tabulated way. After the tabulation, you have to describe it, and also you can put it as graphs. Also you can put it as photographs, micrographs, diffractograms, chromatograms, and all the other types of sophisticated methods we can use. So this is the summary of data collection and display today. So now today we have already come to the last stage of this research methodology module. So hope to see you next in revision class.